Welcome, and recall that last time when we left off we had zoomed in to the cell wall structure of a bacterial cell. So this green layer on Norbert, we were checking out the ultrastructure on it, and so we had another sweet model that essentially if you were zoomed into that green layer, you would see what would look like this or like this, depending upon whether you were looking at a gram-negative or a gram-positive cell structure. But recall that we had further zoomed in, and in fact we had zoomed in to a region of peptidoglycan that if you put a box around this region right here, you would be looking at the region that we have zoomed into on our picture. And this model doesn't even go into enough detail to show the tetrapeptide chain and in gram-positives, the pentaglycine interbridge. But one of the things that we were talking about as well last time was the fact that peptidoglycan is somewhat analogous to a tea strainer. Remember how it allowed water to pass through those pores and easily be permeable, while at the same time providing a great deal of rigidity to the cell wall. So we noted that peptidoglycan is permeable to many substances. These include things like sugars and amino acids and ions. It's somewhat malleable, but it is also a fairly rigid layer that allows for a lot of structural resistance within a bacterial cell. Now recognize that peptidoglycan, while it is the same named structure across all bacterial cells, varies from one group to another. So for example, if we did assume that the gram-positive cell that we're showing in this model is Staphylococcus aureus, and we assumed that the gram-negative that we're showing in this model is E. coli, then we could make note that despite this being peptidoglycan and this being peptidoglycan, the nature of the peptidoglycan would vary just a little bit. And that's what I'm showing you in this picture. Notice that in E. coli, we still see these unusual amino acids. We do not see the pentaglycine interbridge. So the peptide interbridge that we see in Staphylococcus aureus, the 5 glycines, it's not there in E. coli. But we also notice that there's some derivations in the tetrapeptide chain. So whereas E. coli contains generally this diaminopamelic acid, we see a amidated version of glutamate within Staphylococcus aureus. Both are unusual, but they're different. So there is some variation across the bacterial world. But one thing that is universally true, and this is so important. Particularly, I know that Leah is tuning in because you have a lot of interest in medicine, and there are a lot of you who do. There's absolutely nothing like peptidoglycan in us. That's also true of archaea and other eukaryotic life. None of us have anything like peptidoglycan. So I hope that's starting to get your wheels turning, because if humans don't have any peptidoglycan, then if we get a bacterial infection, what is true that we can do to help treat that infection? So I want you to give some thought to that and to think about what are the medical advantages that can be exploited, if you will, within the bacterial cell in order to make a target for these antimicrobial substances. And these include things like antibiotics. For example, penicillin inhibits the enzyme that builds the tetrapeptide chain. In fact, interestingly, that enzyme was named PBP, or penicillin binding protein, for the fact that penicillin inhibits the formation of the tetrapeptide chain in peptidoglycan. Our body also takes advantage of this. So when we cry, for example, in our secretions, in our tears, there's an enzyme called lysozyme. Lysozyme is an immune factor. It allows us to cleave the bonds, the glycosidic linkages between NAM and NAG, and to break down the peptidoglycan in potential gram-positive pathogens. So not only does the medical world exploit this, but our body's immune system recognizes this very important difference between bacteria and us and uses that as an immune factor. There was a great question that came up in my class just a couple of semesters ago. His name was Tucker, and he just wanted to know how is it that antibiotics only manage to kill the bad germs? The tricky answer to Tucker's question is that they don't. And this is wherein lies the problem, or one of the many problems with antibiotics, is that when you do take antibiotics, they also kill much of your microbiota. So many of the bacteria that line your intestinal tract, they're inhibited or killed by those antibiotics, and others of your mucosal surfaces. So many of your good bacteria are also damaged by this. And so some of you have recognized that one of the major impacts of taking antibiotics, particularly over a period of time, is an increased likelihood to get yeast infections. That makes sense, right? Because yeast are eukaryotes, and so they're not affected by the antibiotics. So when your normal populations of bacteria are inhibited by your taking of antibiotics, the yeast are like, woohoo, I get more of the plenty, bring it on, I get to take over. And that creates an imbalance, which creates infection in the form of a yeast infection. Finally, at long last, we're ready to jump into the nuanced structures of the gram-positive and gram-negative cell walls. We've been looking at this model and seeing that gram-positives are distinguished by that thick layer of peptidoglycan. Here, this model shows 9 layers of peptidoglycan. I have another sweet 3-dimensional model that shows 11 layers of peptidoglycan. However, sometimes a gram-positive cell can have as many as 30 sheets of peptidoglycan. So it can become an immensely thick layer that is very highly protective. There are other molecules within the gram-positive cell wall as well that are very distinguishing. In this model, you can see that this student has shown them as the green pipe cleaner and the white pipe cleaner. Together, these are called the teichoic acids, and they are either of one of 2 types. So let me show you in this other model how they are distinguished. You can see here that one of them, called lipoteichoic acid, is aptly named because it attaches to the phospholipid bilayer of the cytoplasmic membrane. So this is lipoteichoic acid here, whereas teichoic acid attaches to the peptidoglycan. Specifically, it attaches to NAM residues. So this model is a little off, showing it attached to the glycosidic linkage when it actually is attached directly to the NAM residue. In both cases, both the lipoteichoic and the teichoic acids are comprised of polymers of glycerol and phosphate. That is, they go alcohol, phosphate, alcohol, phosphate. So phosphate, being an anion, is negatively charged. Wait, what? Gram-positive cells are negatively charged? Yes, they are. So please remember that gram-positive only means that this is the type of cell that retains a primary stain when stained with crystal violet. That has nothing to do with the charge. Just like other cells, gram-positive cells are negatively charged, and that negative charge comes from the phosphate anion. So recognizing the difference between teichoic and lipoteichoic acid, and then recognizing their contribution to negative charge. But additionally, it's cool to think about the fact that these 2 teichoic acids, as you might guess, are also recognizable by our body's immune system. So if a gram-positive escapes into areas that it normally shouldn't be, and begins to cause infection, your body is going to recognize the teichoic and lipoteichoic acids. Said another way, these molecules are immunogenic. They're going to trigger your immune system to react to them. We also say antigenic. That's another term to say foreign invader that stimulates the immune system. So an antigen is a foreign invader. Now, we can look at a different form of a picture, and in fact, this picture is maybe a little bit more like this model, where you can really start to see some of the layering in a more zoomed in way, and you can see the teichoic and lipoteichoic acids. Sometimes this is actually called a periplasmic space, even though formally gram-positives don't have a really large periplasmic space. You will sometimes see other proteins found in this region of a gram-positive. But overarchingly, you might say that the gram-positive cell has a little more simple and more different structure than as compared to a gram-negative. Gram-negatives have an outer membrane, and so because of that outer membrane, they are a little more similar in the external layer to a eukaryotic cell because it's a cytoplasmic membrane that is on the most outer layer. Now, I've drawn a simplistic version or simplistic picture of the gram-negative cell here just to kind of show you that layering and to recognize the difference between the cytoplasmic membrane, which here I'm showing in gray, versus the outer membrane. But if we zoom on in onto that outer membrane, and in fact, let's use our model to help us do this. So here we've got the inner cytoplasmic membrane, and then we've got the periplasmic space here with the thin peptidoglycan. Sometimes it's as thin as one layer peptidoglycan. Here we see three. And then if you look at the outer membrane, and that's where we really want to take our attention now, you'll notice that the inner leaflet is all phospholipids. But if you look closely, you'll recognize that the outer leaflet contains this special molecule that this student has depicted with a square with kind of a fuzzy thread on the end. This very special molecule is called lipopolysaccharide, or LPS for short. Lipopolysaccharide is immensely important in a gram-negative cell for a number of reasons that we're just about to discuss. But do take note that it is comprising a big portion of the outer leaflet of the outer membrane. Now, that outer membrane encases and protects the peptidoglycan of a gram-negative cell. So it's difficult for substances to get in and attack the peptidoglycan. So if you think about whether or not a gram-negative cell would be inherently susceptible to penicillin, it's unlikely because the penicillin would have to get through this outer membrane in order to get to the target, the peptidoglycan. So the cytoplasmic or the outer membrane protects the peptidoglycan of the cell, and that peptidoglycan is nestled in the periplasm. So the periplasm, remember, being this region here between the inner and the outer membranes, or between the cytoplasmic and the outer membrane. So now that we've looked at a simplistic depiction of the gram-negative cell wall, we can look at some more complexities. And in fact, I want to zoom in to the lipopolysaccharide molecule, and I want to actually look at the ultrastructure of the LPS molecule. So if we literally zoom in and we look at this region here, which is the hydrophobic tails of LPS, and then we look at the core region and then the saccharide chains that are shown with the threads, that's what we're going to zoom in on next. Specifically and interestingly, this is the lipopolysaccharide molecule from Salmonella, which I find that super interesting because one of the vaccinations that I need to get for Kazakhstan is a vaccination against typhoid, which is Salmonella typhi is the bacterium that causes that disease. Now, Salmonella is a very smooth criminal, and its LPS molecule is one of the things that allows it to be a smooth criminal. Recognize that this topmost portion of the carbohydrate region of the lipopolysaccharide, or the polysaccharide region of the lipopolysaccharide, this O side chain is capable of changing. So you could think about this as the Salmonella mask. It could change its mask, right? Put on disguises, and by changing that O side chain, it can confuse our immune system, and our immune system thinks, wait, that's not the criminal I was looking for, and it gets very confused by that. So that O side chain has that function. It also has other functions as well. It is immunogenic too, right? Obviously, if it is recognized, if the immune system can find it, if it doesn't change its mask and it hones in on it, then it can cause the immune system to be triggered by it. So it can also play a role in adherence to surfaces, and we'll talk about that momentarily. But first, I want to draw your attention to where the gram-negative cell gets its negative charge. Notice that there are phosphate anions that are attached to the core polysaccharide region of the LPS molecule. And then if we go all the way down to the very bottom of LPS, this is where we see the lipo part of the lipopolysaccharide. This is the lipid part, explicitly called lipid A. So lipid A is embedded in the lipid bilayer and it stabilizes, but it also is toxic. So in fact, LPS is an endotoxin, meaning that get this, if you, and there's a number of you who are really passionate about agriculture. I think Thomas might have been one of the people that said that. If you took LPS alone, all by itself with its lipid A component, and you inject it into cattle, they will get incredibly sick. They will go through fever and shock. And that is strange, right? Because there's no offending bad guy. It's just a molecule, but the body thinks it's been invaded. So the lipid A is toxic. It's an enterotoxin. It's causing your body to recognize it and think it's been invaded. So combination between the lipid A and the O side chain says, whoa, criminal has entered the stage. Now, I want to make note that if you're a bacterium, you look at your lipopolysaccharide and say, man, this molecule is the shiz-niz. It allows me to do so many cool things. One of those things that it allows me to do is to escape defense systems. But another thing that it allows me to do is to adhere to surfaces. And in fact, it enables gram-negatives to form a sweet biofilm. Now, a biofilm is defined as a complex slime-encased community. So, catch that, I'm going to restate it. A complex slime-encased community. So it's very complicated, and most biofilms are comprised, they're polydiverse, meaning there's more than one microorganism that lives in them. Remember seeing my kombucha biofilm that I showed you at home? So these are really complex, very strong and stable communities, and they tend to be very resistant to things. They're very much a boon to the bacterium. You allow it to live in complicated environments. So this LPS molecule in the outer leaflet of the outer membrane very much creates a permeability barrier. So we mentioned this, that things like antibiotics can't get through the outer membrane very well, partly due to the LPS layer. Other things like biosalts, other toxins, hydrophobic toxins, can't get in very well. Now, this makes sense actually if you think about it. Gram-positives tend to live in our nasopharyngeal area and on our skin, whereas gram-negatives tend to live in our gut. Now, there's some exceptions to that, but it's a good rule. And where do we find bile? Right? It's what allows us to digest fat. It's the detergent that allows us to emulsify fat in our digestion. So it makes sense that something that lives in our gut would need to be pretty resistant to that. Very cool. Now we can zoom on in and look at all of these parts in a different kind of model and maybe go ahead and label them here, the lipopolysaccharide, the periplasmic space. And I'll draw your attention now to a couple of important molecules that might be easy to overlook. The first one is the large brown protein. This is a pore. In fact, it's called a porin. And it allows pretty large molecules to get into the outer membrane. So unlike the cytoplasmic membrane, the outer membrane is a little less exclusive. It's not quite as restrictive about what gets in as the inner membrane is. The porin proteins will allow anything under about 600 to 700 Daltons to diffuse in. Now, what does that mean? I mean, sucrose table sugar is about 323, so it could just swoosh right in, right? A disaccharide like that, no problem. So amino acids and other things along those lines. I also want to draw your attention to something called lipoprotein, and or aptly, this is also called bronze lipoprotein. And this molecule literally knits together the outer membrane with the peptidoglycan of the cell. So it plays a very important role structurally. We'll go ahead and jot a bit of that down. Lipoprotein joining outer membrane to the peptidoglycan layer, and porin proteins forming pores through which small molecules can pass. This is an image of a porin protein. You can see that it is a beta barrel. Very cool, right? See all that beta secondary structure that exists in that transmembrane or integral membrane protein. Let's think about a bacterium named Klebsiella pneumoniae. This is not usually a pathogenic bacterium, but it can cause, if someone is immunocompromised, it can cause pneumonia in those people. So we want to know which of the following would typify the cell wall of this bacterium. So notice that it is gram-negative. So perhaps one of the first things to draw your attention to as a part of the question is just that this is a gram-negative bacterium. So we know that it's going to be typified by all of those components that we just talked about. And we can maybe find or select some of those components. But first, let's just think about answer A and whether or not it would have a thick layer of peptidoglycan. Well, remember that that wouldn't be true because it would have just a thin layer. So remember the very thin layer of peptidoglycan there. So we can cross off A. Lipoteichoic acid, that can be crossed off as well. And that's because lipoteichoic acid, remember that that is the polymer that joins or that is bound to the cytoplasmic membrane and extends off of gram-positive cells. So in fact, we can turn this model around and we can find lipoteichoic acid on the gram-positive side. So here we've got the lipoteichoic acid attached to the cytoplasmic membrane and extending from a gram-positive cell wall. Next, lipopolysaccharide, absolutely, right? LPS is that major molecule that constitutes the outer leaflet of the outer membrane of a gram-negative, and like all cells, a negative charge. So the answer that we come up with for this question is F. C and D would both be totally rad answers. I want to make a note that some of you might be craving some conversation. I can hear Thomas right now thinking, how do archaeal cell walls differ from bacterial cell walls? That is, Rachel said that only bacteria have peptidoglycan, but we know that archaea also have cell walls. How do they differ? Well, one note on that is that they don't have N-acetylmuramic acid. They don't have NAM. So they can't make a peptidoglycan molecule that would be equivalent to what we see in bacteria. They have a molecule called pseudomurine that has sort of a somewhat structural similarity, and they can build cell walls. So later when we talk about archaea, towards the end of the semester, we'll do a survey of them, and we'll actually do a virtual field trip to Yellowstone, so that'll be really awesome. And we'll go ahead and talk about more about the structures of the archaeal cell. So you can anticipate that, breathlessly anticipate that, shall we say. I'd like to talk now, though, a little bit about a concept that I know you've been exposed to before. This is osmosis. You know, we have that sort of, I don't know, what is it? It's like the urban use of the term osmosis, where we think, yeah, if we sleep on top of our book, maybe it'll come in by osmosis. So while that's a sort of urban dictionary use of osmosis, it is not the technical definition of the term. Instead, what osmosis means is the movement of water. So the movement of water in response to solutes. Water always goes from a concentration of lower solute to higher. That is, when we look at a cell, and on the left, upper left here, we see this, looks like gram-positive bacterial cell, where we see a thick peptidoglycan layer, we see a cytoplasmic membrane, and notice that inside of the cytosol, there's a higher concentration of solute than outside of the cytosol. So water, in this case, is totally going to rush in to this cell. And when it rushes into the cell is going to bulge. And you might be thinking to yourself, the cell is going to burst. But what prevents the cell from actually literally bursting? Remember that rigid peptidoglycan, thick in a gram-positive? It's just going to push the cytoplasmic membrane up against that peptidoglycan. So the cell wall tends to help the cell from bursting as water rushes in. So let's talk terminology here. If the solute concentration outside the cell is lower than inside the cell, we say that the environment is hypotonic. Hypo means less than. So that term, that prefix, hypo, means less than. So there's lower solute concentration outside the cell than inside, so it's a hypotonic environment. We know that that cytoplasmic membrane gets smushed up against the cell wall, And this actually is somewhat enjoyed by bacteria. They actually kind of like it. Um, it gives a little bit of turgor pressure, and they kind of like that just taut membrane up against the cell wall. Now, what if this were mycoplasma? And I think a couple of you had walking pneumonia. If you've had walking pneumonia, remember that that's caused by mycoplasma and it lacks a cell wall. If mycoplasma had too much of a hypotonic environment, then we would see that that cell would burst, right? So that's something that is of consideration, is whether or not that cell wall helps to keep the cell from bursting in a hypotonic environment. And therefore, the reason that most bacteria enjoy a hypotonic environment, but some of them don't. Let's look at the opposite scenario. And I am curious if any of you have ever done any making of beef jerky and preserving the meat through the formation or the salt treatment of the meat. This is all about osmotic pressure. So the preserving of meat through curing with salt is because of the opposite environment than we looked at before. That is, putting a ton of salt around a cell creates a hypertonic environment. And in a hypertonic environment, the water is going to rush out of the cell. And the cytoplasmic membrane is going to shrivel. And we call that plasmolysis. So as the water rushes out, the cell membrane shrinks. It pulls away from the cell wall. And this is called plasmolysis. The rough thing about a hypertonic environment for a cell is that cells don't have any protection against plasmolysis. And in fact, in a very hypertonic environment, they die. This can be a really good thing, right? Because when we preserve meat using salt, that actually is a very effective way to kill bacteria. So salt preserved meat, beef jerky, can sit out on the countertop for long periods of time and it doesn't go bad. And that's because the bacterial cells that might have contaminated it have undergone plasmolysis and they've been killed by that hypertonic environment. So if you would like to email me for extra credit, a little bit about experiences that you might have had with hypertonic environments and how they can preserve meat. Or maybe you've been in a lot of you like to travel. I know, related to me, that passion for traveling. Maybe you've been to a place where the use of a hypertonic environment is commonly used in preservation. Tell me a little bit about that, and I would love to receive your emails. Do know, however, and you can email through Canvas. Do know, however, that I am a bit behind on my email, so it may be a while before I get you extra credit, but I promise that I will do so. I want to talk about a couple of lingo, sexy terms. These are protoplast and spheroplast. A protoplast is quite literally a cell without its wall. So if we have a cell that lacks, for example, if we've got a gram-positive cell and it completely lacks its thick peptidoglycan, we would call that a protoplast. That is, it would be nothing but the cytoplasmic membrane and no cell wall. A spheroplast is where the cell wall is partially degraded, though not entirely degraded. So coming back to our extra credit, what I want you guys to also tell me in your email, and both things for full credit, or for credit. So I also want you to tell me how we might make a spheroplast. That is, how might you take this gram-positive cell and weaken the cell wall, making a weakened cell wall or a spheroplast. That is what chemical that we talked about could be used to do that.